Today we are going to learn about codominance. In the previous class we learnt about the incomplete dominance. Incomplete dominance and codominance, they worked against the universal application of Mendel's first law, that is Mendel's law of dominance. They were actually the exception to Mendel's first law of dominance. Codominance is the phenomena of inheritance in which the two alleles of a gene express themselves together, that is simultaneously and in the presence of the other. This means that both the alleles are equally dominant. The F1 progenies in codominance, it resembles one of the parent, which is in contrast to Mendel's first principle, that is law of dominance, in which the F1 progeny resembled one of the parent, that is all the progenies were dominant. The well-known example of codominance is the ABO blood group of human beings. The ABO blood group of human beings. Human beings, they have 4 different types of blood group, that is A, B, AB and O. The blood group character is controlled by gene I. Which is present in 3 allelic forms. They are IA, IB and I. Now this IA, this A and B, they are actually glycoproteins or sugar polymers which are found attached to the surface of the RBC. The allele IA, it produces glycoprotein A. The allele IB produces glycoprotein B, and recessive I, this is recessive, it has no glycoprotein, it does not produce any type of glycoprotein. So this is recessive. IA is recessive to I and IB is also recessive to small I. IA and IB are codominant. That means that they show their presence simultaneously in the presence of each other and they are equally dominant over each other. Hence, the RBCs have got 2 types of glycoproteins, A and B. Although there are 3 different allelic forms, but every individual possesses 2 of them. We can make 6 different combinations of genotypes from these 3 allelic forms. Let us see the table. Now this is genotype and phenotype, or we can say the blood group. IA and IA, it belongs to blood group A. Both are dominant, IA and I. So this will be also A, heterozygous A. IB and IB, this is B, B, I, this will be heterozygous B, and then another we have got is A, IA and IB. This will be AB. This is blood group without any glycoprotein, so it will be blood group O. So here we see IA is dominant. This means it belongs to blood group A. An individual will have blood group A. If it is heterozygous, IA and combination with recessive I, then the blood group will also be A, but it will be heterozygous. Now IB and IB dominant, the blood group will be B. IB combined with I will be B also, but heterozygous. Now IA combines with IB, both show codominance. Both are showing codominance and the blood group will be AB. The individual will have the blood group AB. If I combines with I without any glycoprotein, the individual will have blood group O. Blood group O is known as universal donor. Now this blood group A is donor, universal donor. Blood group AB showing codominance. It has both A and B. So it is universal acceptor. The blood group ABO also shows multiple allelism. So we can say that blood group ABO of human beings, other than depicting codominance, it also shows multiple allelism because it exists in 3 allelic forms, that is IA, IB and I. Bernstein in the year 1925 discovered multiple allelism and codominance. That is, he discovered codominance, and multiple allelism in human blood. Now let us know what is multiple allelism. Multiple allelism is a phenomena in which a gene exists in more than 2 allelic forms, and an individual can have only 2 of the allelic forms. Therefore, multiple allelism can be studied in population but not in any individual. Now let us again recall. We see that blood group A and blood group B can be homozygous also and heterozygous also. Blood group AB is always heterozygous and blood group I, that is O, is always homozygous recessive. The knowledge of blood group is always helpful, and we all must know the blood groups to which we belong. It not only helps us during emergency, it also helps in solving the parentage problem.